Okay, so this is chapter 6, basics of metabolism, basic concepts of metabolism. Okay, so far I've only learned about two metabolic pathways, which are catabolic and anabolic. So catabolic, think of it as cataclysm, the one from Ladybug and Cat Noir. Cataclysm means breaking things. Breaking things need more energy. For example, you wanna break a healthy couple. You need more energy to do that, more effort. And for anabolism, for making a beautiful couple, I don't think you need to put much energy in it, because beautiful things just happen, I guess. So there's that. So catabolism is a convergent process, which means wide variety of molecules are transformed into few common end products, right? So that means a lot of when you have little things, there are more variety in the little things compared to let's say the bigger things. Okay, you can only have 3 big things. Let's just say okay, in a bag you can possibly fit, let's just say, 2 big things. But in the same bag, you can possibly also fit 20 little things. Okay, so you understand the 20 things means more variety, whereas the 2 things, it means less variety. So for convergent means that. So cataclysm, convergent, remember this. And anabolism is divergent. That means a lot of a wide variety of products can form very few end products, which are complex and polymeric. Okay, so for catabolism, we have 3 stages. Let me write them down. Okay, so as you can see, the first stage is the hydrolysis of complex molecules. So complex molecules being proteins, polysaccharide as in the complex form of carbohydrates, and lipids. So when you put water into them, you know that they break down into their building blocks, which is amino acid, monosaccharide, and glycerol and fatty acid. Okay, this whole thing is catabolism. Okay, when you go this way, it's anabolism. When you go this way, it is catabolism. Okay, so what happens in stage 2? Stage 2 is when these building blocks all form acyl CoA. I have no idea what CoA means. Don't ask. Okay, so what happens here is these building blocks turn into acyl CoA, and which in turn, in stage 3, which is oxidative phosphorylation, participates in the citric cycle and of course forms the basic forms of energy and byproduct, which is ATP and carbon dioxide. Okay, so now we're going to talk about regulation of metabolism. So there are 2 ways metabolism is regulated, one of which is signaling from within cell. So the cell itself signals, okay? Or cell-to-cell communication, which is passed along the cell, to remind you how, metabolic rates are just regulated. For example, okay, let's just say right now, your cell, okay, it has a lot of glucose, okay, that it can't digest. I don't know, is insulin used when there's no digestion? Okay, anyway, please answer that if you know. Okay, so yeah, let's just say there is this is a very made up example, so please do not refer to it. Okay, so the cell itself signals about the metabolic pathway rate, okay? Or the cell tells the cell beside it that, hello, I've got a lot of glucose, Can you just like pass the message along? So it's like that, okay? So let's talk about this. The cell doing it within itself. I'm sorry for the lighting. I'm just going to read it out loud. Cells from within cell, signals from within cell increases if the rate of metabolic pathway increases. Okay, I just use this to just, doesn't make sense, but it does. Okay, so yeah, there's that. It increases, this also increases. Yeah, it's like, you know, if you have a lot of metabolic rate, like if your body's really working good, you're gonna do it by yourself. Okay, you don't need to just like communicate through someone else, right? It's like that. So yeah, like that. So there are some factors that influence the metabolic rate. This being availability of substrates, the amount of substrates present. Let's just say, you know, this example that I used before, that we have a lot of insulin. Let's just say one of my cells has a lot of glucose, right? So let's just say that compared, glucose is a substrate. Glucose is a substrate. So there's that. A lot of glucose. Product inhibition. Let's just say, I need another example. Okay, you know those enzymes? No, not enzymes. Those products that inhibit their own reaction. Like let's just say you want to make ammonia. This is not one of those examples, okay? Google this. I'm pretty sure this is not that example. Ammonia, okay? Let's just say you want to make, okay, no, this is very risky. You want to make product C, okay? A plus B equals to C, right? This is X enzyme. So let's just say you're making a lot of C, right? But you also have a lot of A and a lot of B. But of course, your body doesn't need a lot of C, right? So your C at times, this is like a specific reaction, okay? Your C works in a way that it inhibits the work of X. Get it? So that is product inhibition. This also, if this increases, rate of metabolic pathway decreases. If availability of substrates increases, rate of metabolic pathway increases. Should just eta. Okay, we're thinking of this being high. Okay, and alterations in activators and inhibitors. Okay, so let's just say the same reaction. There is another thing, Y. Okay, now this isn't the one which is inhibiting the reaction. There's something else called Y. So what happened is, so this is your inhibitor, which is inhibiting this thing. So let's just say after one point, because of temperature, let's just say this is an exothermic reaction. So because this produces a lot of heat, this might get altered. Hence, this will increase. And, or let's just say there was Z, something like Z. Now this is an activator, as in this activates this enzyme, right? Now, for some reason, once again, this is an exothermic reaction. This might just, you know, raise the temperature, and it might, what is the word for it? It's called, like, you know, unshape the molecule of Z, so it might not work as an activator anymore. So that's why the rate of metabolic pathway will decrease. So it's all in that. So that is signals from within cell. Now let's talk about cell-to-cell communications. For cell-to-cell communications, there's indirect and direct. So direct being like that. Duh, like that. Okay, like, you know, the cells are just together and bro's just like whispering onto his or her bro's ears that, bro, there's something wrong with the reactions that's happening inside my body, like the metabolism. So like just inform it to the next person. Okay, that is cell to cell link. Very clingy. Surface to surface contact. Okay, and then we have indirect. So the thing with indirect is, okay, we're bros, but we're not as clingy. So we just like signal each other, like, you know, situationships signal each other like that. So that is done via hormones or neurotransmitters, because, you know, you get that little thing and there's a receptor, and then that does that, and then that just does that. We're gonna get into this. Okay, so let's talk about transduction. Okay, so the thing about transduction is just a fancy word for chemical signaling, I guess. Okay, I'm not sure myself. I wrote something down, but I can't read my own handwriting. So, you know, there is that. So, okay, so there are two types of transductions. There are cell surface receptors and intracellular receptors. Now let's talk about cell surface receptors, because I kind of did the whole thing here. It only focuses on the cell surface, okay? Like the chemical, the things that are signaling. For example, you know, your brain, okay? You touch a hot pan because you're a dumbass, right? So your brain is telling you, okay, that is it gonna be like that? Okay, just like go with it. Okay, go with it. Okay. Who cares if the example is valid or not? Okay. So your brain's gonna tell you, okay, dude, take your hand off, right? So now it's gonna signal all the way from here to here, right? So the whole thing is gonna go here, all the way here. Acha. So this thing that is signaling your brain, which is a neurotransmitter. Okay, we're focusing on this one right now. Neurotransmitter. It's gonna attach itself here. Okay, and this is an ion channel for now. We're gonna assume that this is an ion channel for now, and this is a neurotransmitter, like this thing, right? And then this thing opens, or like something happens, okay, and then, you know, you do your thing. So you understand that this does not enter the cell. That is cell surface receptors in transduction. Okay? Now, let's talk about this, catalytic receptors. Now, you've had a lot of sugar, right? A lot of sugar, right? Now, let's talk about cell-to-cell signaling, the thing that we did a little while ago. Okay? Your cell next door just told you, okay, it has a lot of sugar. Okay, it needs to like just pass down a message that, you know what, I have a lot of sugar, I need insulin. So this thing, once again, imagine this is the text message that tells this one that was a really bad example. Sorry. I'm so sorry. Do not forget I said that. This is a catalytic receptor, right? So imagine that there are hormones here. There are hormones already present in your cell, okay? But we are not using it. We're not using it, okay? So what your receptor does, okay? Your brain sends signal to your thing. I think there is one thing with kidney, ADH or something like that. Okay, anyway. This thing, it attaches here. Wow, okay. It attaches here, and this thing just like does the whole catalyst thing in here. See, it's still in the surface level. Get it? But except this time it's for catalysis. catalyst. Okay, then we have second messenger. This one is a little complicated. I will be going into detail by the end of this video, so just think of this one as, yeah, I mean, it doesn't really go there. Okay, you know what, let's go with it. Imagine there is another thing here. Okay, so imagine that there is this molecule thing here, right? Something attached itself here, which is some signal. Okay, some signal from here. So upon that, this thing comes all the way here and attaches itself to this, and it changes shape. So this box will be called a second messenger. Get it? I think that was a really bad explanation, but we just gotta fake it till we make it. Then, so let's talk about this one, which is honestly the thing that I actually understood. Okay, so imagine a really good example of this is, for my skin girlies here, retinoic acid. We know what retinol does, right? It's an active ingredient. It actively works on your skin to just anti-age and all that stuff. So you know, all this anti-aging stuff and all that stuff, it really works on your DNA, right? There's like no chemical thing happening. That's mainly your genetics that we're really trying to work on, right? Also vitamin D. Okay, why do we really tan when we're exposed to a lot of vitamin D? Okay, so these stuff are actually called I don't know what they're called. Okay, so these things actually happen in your cytosol or the nucleus. Okay, so vitamin D. Let's talk about vitamin D. When you go out in the sun, you get a lot of vitamin D, and you get tanned. You get tan. So why do we get tan? Now, if you know the whole thing about melanin and stuff, your gene that is responsible for tanning, that is expressed a lot. I'm not sure if I'm going the right direction, but bear with me, all right? I'll be saying a lot of nonsense. Okay, so what happens is the receptor in your cell, ow, that hurt. Okay, the receptor in your cell, okay, so this is your receptor. Why are we doing it here? This is your receptor, okay, and this is your nucleus, right? This is your DNA. Makes sense, right? So this thing will come and attach here, or this thing will enter here, okay? Or maybe it'll do its work here, right? Either ways, it's gonna come here inside this, and somehow, I mean, it has a direct effect on your DNA. Okay, so these things, okay, these receptors, these are called intracellular receptors, okay? So these enter the cell, okay? But there are a lot of chain reactions that occur, after which, one of the things, maybe it itself or something else, it causes something else to bind with one specific region of DNA. So, one trait is heavily expressed. Get it? Please leave any questions below if you have any, although this video is made for self-revision purposes mainly, but if you have any questions, please put it down in the comments. If I have miscommunicated anything, mispronounced anything, made a mistake anywhere, please forgive me, and please note it down in the comments. Thank you so much. I will be covering intracellular receptors, transduction, the thing in detail, like sort of in detail, in the next video.